everyone. My name is Cameron, and I would like to welcome you to the first Joy of Meiosis series. If this is your first time with me, allow me to extend a warm welcome to you, and encourage that you use a pen and pencil to take notes on this excellent discussion. Now, with meiosis, it is very similar to something that you might have learned earlier called mitosis. Now, there are two major differences between the two. In meiosis, cell division occurs twice, which is opposed to mitosis where it only occurs once. And the byproduct of meiosis is 4 cells, compared to the 2 cells that occur in mitosis. The 4 cells, however, are made up of 23 chromosomes. The cell name of a 23 chromosome cell is a haploid cell. That was a little bit of a tongue twister there. For the male, these are called sperm cells, and for the female, they are called the egg cell. Now, when cell division occurs, in both mitosis and meiosis, interphase needs to occur. In interphase, DNA is replicated. So as you can see, I have 2 green veggie straws here. DNA duplication allows for me to add 2 green, 2 orange, and can you guess what else I'm gonna add? That's right, I'm assuming you got it. Now, we have 92 total strands here of what it would be. 92 individual strands of DNA. Now, only here I'm going to be using 3 pairs. And while this is not what happens in a cell, I'm sure you'll be able to adapt. At least I hope to the circumstances that you were able to watch. Now, in the prophase, my bad, I had my words tied up there. Chromosomes begin to form, which are made up of 2 chromatids each. You can see they are represented by the individual colors here. These pairs can be referred to as homologous pairs, as they will be the ones that are mixing with one another in a phase called crossover, which we'll be delving into right now. In crossover, the two sets of chromosomes are bounded with one another. And unlike in mitosis, this is what causes all of us to be individuals and not like clones, which are produced in mitosis. Individual genes that are a part of the chromosomes are transferred with one another, causing there to be 4 completely different chromatids. This is what allows for me to be different from my brother and not an exact copy. Because if mitosis were to occur when making haploid cells, we would have non-existent differences from our siblings and our parents. Now, next we have the metaphase. And unlike mitosis again, meiosis actually pulls apart the 2 sets of chromosomes instead of chromatids. They are lined up in the middle in metaphase, which can be determined by the M, which is middle. It's not actually middle, but I just like to add a little flair to it.
Hello, my name is Abby Grace Kimsey, and I would like to welcome you to the Joy of Anaphase 1 and Telophase 1 in the Meiosis series. If this is your first time with me, allow me to extend a personal invitation for you to get your pens and pencils and take notes along with us in this discussion. Anaphase 1 is the third stage in meiosis 1 and follows metaphase 1. During anaphase 1, the homologue chromosomes are pulled apart to opposite sides of the meiotic cell. The homologues are pulled through the spindle apparatus, which forms at both ends of the cell. During anaphase 1, the sister chromatids stay connected to one end of the other and don't separate. Sister chromatids are two copies of the same chromosome formed by DNA replication attached to each other by a structure called the centromere. A homologous chromosome are two pieces of DNA within a diploid organism which carry the same genes, one from each parental source. Each homologous chromosome can provide a different version of each gene, creating more genetic variety. In telophase 1, the fourth and final step in meiosis 1, chromosomes arrive at opposite poles of the cell. The spindle breaks down and the chromosomes are bound in a new nuclear membrane. The chromosomes then uncoil and form chromatin, the material of which the chromosomes of organisms other than bacteria are composed. It is made up of proteins, RNA, and DNA. These new nuclear membranes and the formation of chromatin leave the cell ready to undergo cytokinesis, which separates the cell into two daughter cells. A daughter cell is a cell that results from the separation of a single parent cell. After cytokinesis, each daughter cell has one set of chromosomes and is also a haploid.
Hey, hello there, and welcome to the Joy of Meiosis. Now, today we're picking up where we left off last episode, and we're looking at the second part of meiosis. Now, here, it's very similar to mitosis, except the aim is to pull the sister chromatids apart into separate, stranded chromosomes, almost like one chromatid each. All right. Now, let's get started. Our first beautiful little phase is almost not even a phase at all. It's called interkinesis. This exists directly after the first part of meiosis. Now, this is almost an instant, very brief, so quick that it doesn't even hardly occur in some organisms. The DNA is in the form of the partially compacted chromatin. Now, we here see that within our prophase, the centrosomes have moved to each of the opposite sides, and we can see that the chromatins come together and compacted to form chromosomes. Now, let's see what happens in our next step. Beautiful. Here in metaphase 2, we see that the centrosomes have moved to either side now extend their microtubules to the chromosomes that have moved to the metaphase plate, otherwise known as the middle of the cell. From here, these microtubules will pull them back into our next phase. Magnificent. Here, in anaphase, we can see that the formerly sister chromatids within each chromosome have been separated and pulled to each individual side within each cell. Now, follow me. Wow, truly something to behold. But how did we get there? Well,
the unduplicated chromosomes from the last step, anaphase 2, have unraveled and turned back into these spools of chromatin. Now, we can see that a nuclear envelope has formed around each side. Subsequently, here we can see cleavage begin to take shape as we move into our next step. My goodness, truly remarkable where a brush can take you. Here we see that where the cleavage began, the cells, which had developed a new cell membrane around each individual cell during the cleavage, have moved apart, and thus we have painted cytokinesis. Subsequently, the two successive divisions of meiosis yield 4 haploid nuclei, each containing one of each kind of chromosome. Each resulting haploid cell has a different combination of genes. Now, let's talk about what brings us all together. Happy little accidents. Another thing I like to call genetic variation. Now, we've observed 2 main sources of this throughout our time here in meiosis. The first, DNA segments are exchanged between maternal and paternal homologs during crossover. The second, we see that the maternal and paternal chromosomes of homologous pairs separate independently, and chromosomes are then shuffled so that each member of a pair becomes randomly distributed to one of the poles at anaphase 1. Now, you must be asking yourself, other than such a beautiful painting, what is the result of all these happy little accidents? Well, I'll tell you. There are about 2 to the 23rd, or about 8 million possible chromosomes as a result of all this genetic variation.
Hello, my name is Maggie Corbett, and I would like to welcome you to the first Joy of Meiosis series. If this is your first time with me, allow me to extend a personal invitation for you to get your pens and pencils and take notes along with us each discussion. Join me as I present the joy of sexual reproduction as seen in meiosis. First off, sexual reproduction in meiosis is portrayed by the fusion of haploid sex cells forming a dipole zygote. An important key factor to remember is that in order for gametes to form, meiosis must occur first. Meiosis goes straight to gamete production in animals, and it also should be noted that this can occur in other organisms as well, but not as often. The formation of gametes is called gametogenesis. Gamete cells develop when germline cells undergo meiosis. Here you can see the diagram represents that. Here's a better up-close look at this diagram so you can visualize it for yourself at home. Following that, male gametogenes are called spermatogenes. These create 4 haploid sperm cells that enter each other's meiosis. Female gametogenes are called oogenes. These form a single cell egg called ovum, which enters meiosis. Pardon my scuff mark here. Meiosis always occurs at some point in a sexual life cycle, but doesn't always automatically precede gamete formation. This is important to note because this often occurs in fungi. Gametophytes produce gametes by mitosis. Female and male gametes are fused together, thereby creating a diploid zygote. When this occurs, the zygote splits automatically, creating a multicellular spore fact. That's all for today's video on the first Joy of Meiosis series. I hope you've enjoyed it. Thanks for watching.